Great. Well, I think that we're going to get started. Thank you all for joining us today at the Connect to Science seminar series. A couple of quick reminders before we get started. First of all, please check out the website that is listed here on Doug's title slide. You can find the upcoming speaker schedule at that website and also get additional information and Zoom links to future talks within this series. Also, during the course of the presentation, if you have a question, just enter it into the Q&A box at the bottom of the screen instead of the chat box. That just helps us to better organize and ask the questions to Doug at the very end. So with that, it's my great honor to introduce our speaker today, Professor Doug Green from St. Jude Children's Research Hospital. Doug probably doesn't even really need much of an introduction. He's one of the most highly cited, most prolific scientists of our time. And many of you are already familiar with Doug and his work. And that's probably most evident by the fact that on his CV that he sent over just a couple weeks ago, he was an author on over 590 publications. And I'm guessing that in the 2 weeks since we got that CV, he's probably already cracked over 600, considering his extremely prolific rate of publications. I think my favorite thing about Doug, and really the reason that I asked him to come and present today, is that he's a fearless researcher. He's one of the few people that can integrate concepts across multiple fields and come up with completely new, innovative ideas that then take his research to the next level. It's absolutely staggering to watch, and it's so much fun to hear about when he presents around the world at some of the most interesting and fun conferences that I've ever attended. He's been recognized for this kind of talent and all of his other incredible successes with awards such as the NCI Outstanding Investigator Award, the Wilbur Cross Medal from Yale University, where he received his bachelor's and doctorate degrees in biology, and also by recently becoming a member of the National Academy of Sciences. I wanted to keep this short so that we could hear about your latest science, Doug, and we're just so glad to have you here today joining us. And with that, I'll turn it over to you.
Thanks so much, Chris. It's really a pleasure. I'm sorry it's virtual, but maybe that means I get to say hello to people from around the world, and welcome. This is really great. In the next couple of minutes, I'm going to teach you all you need to know about autophagy to follow this talk. Assuming, of course, I can make this work. Great. All right. So autophagy is present in all living things. And again, thanks everybody for your patience. All living things have this mechanism. What autophagy does, what it almost certainly evolved to do, is to keep cells alive when they're faced with starvation, when their nutrients in the environment are not sufficient. And it allows cells to persist until those nutrients become available. The way it works is that under conditions of nutrient deprivation, when the AMP levels increase to greater than the ATP levels, that engages a kinase, AMP kinase. In addition, as amino acids become unavailable, another kinase, the TORC1 complex, mTOR and the TORC1 complex, becomes inactivated. And either or both of those signals coincide to regulate a complex of proteins that sit on the endoplasmic reticulum facing the cytosol. And together, the components of that complex function as, as a serine kinase. Now, this serine kinase does what serine kinases do, and all of the components of that complex are required for this kinase activity in response to nutrient deprivation. This kinase now phosphorylates substrates. That's what they do, and the next set of substrates are present also on the cytosolic face of the ER. And together, the components of this complex function as a PI3 kinase, a lipid kinase. This is not the PI3 kinase you know in cancer biology and immunology. Those are class 1 PI3 kinases. This PI3 kinase is a class 3 PI3 kinase. It's the only one that exists in us. Its function, it generates a slightly different phosphoinositide, a PI3P, which is now decorating that ER membrane. That phosphoinositide now acts to recruit bridge molecules. And one bridge molecule in particular binds to that and recruits another complex of proteins, which together function as a ligase pathway. Now, at a deep molecular level, this is very much like the ubiquitin ligase pathway. There's an E1, an E2, and a rather complex E3. And in the ubiquitin pathway, of course, these function to pass a small molecule, ubiquitin, through the pathway to ligate it to targets, which in this case are proteins in the ubiquitin pathway. In this pathway, in the autophagy pathway, the small proteins are a family of proteins. I'm going to call them LC3. This family of proteins, there's about 6 of them in us. These are transferred via the ligase pathway to conjugate not to a protein, but to a lipid, phosphatidylethanolamin, and also to some extent phosphatidylserine. So now LC3 is lipidated on that ER membrane. Now LC3 binds lots of proteins that have specific features that help to coordinate the effects of autophagy, but among the proteins are motor proteins. And the motor proteins now drag the LC3 out together with the membrane, and that ER sheet will now fold over itself and now in an LC3 dependent manner fuse with itself to create a double membrane vesicle called an autophagosome, trapping cytosolic materials. Again, in an LC3 dependent manner, this autophagosome, this double membrane vesicle, will fuse with lysosomes, digesting the inside, the contents of this vesicle, which are organelles, proteins, other bits of cytosol, either directed or random, and it degrades them down to its core metabolites to provide energy and building blocks to keep the cell alive. That's autophagy. So if you just remember the serine kinase complex, the lipid kinase complex, and the ligase pathway, it's all you're going to need to know. And this idea of LC3 lipidation. Now, our story is going to start with Larissa da Cunha, who was a postdoc in the lab now about a year and a half ago. She left to return to Sao Paulo in Brazil to set up her own lab. I can highly recommend having Brazilian postdocs for a number of reasons, but among them is the fact that they invite you to come and visit, and it's a great place to go. So highly recommended. Larissa was a wonderful postdoc, and she wanted to understand the function of autophagy in the tumor microenvironment. She started with a really simple tumor model. At the time she started, this wasn't really a model that most people would consider a cancer model. It's a B16 melanoma that you grow in culture, you inject them intravenously, they colonize the lung, and a couple of weeks later, you can look in the lungs and see those colonies forming. You can count them and quantify this. I said, well, we'll have to certainly move to other tumor models, which we did, and I'll show you a little of that. But in between the time she started and when she went to submit the paper, checkpoint blockade therapy came along, and it had actually been discovered using exactly this kind of model. And so suddenly, it was okay to publish on this model of cancer. But as I'll show you, this applies to others as well. She was particularly interested in the role of autophagy within the major cell type in the tumor microenvironment, the macrophage. Now, macrophages have, all animals have macrophages. They have different functions. And those functions emerge depending on the signals they receive. So macrophages in a infectious environment, macrophages, for example, that have sensed the presence of bacteria or viruses, will polarize towards an activity which is sometimes called an M1 type macrophage. It's involved in host defense, but this is also the macrophage that can cause tissue damage. It's the major macrophage in inflammation and inflammatory disease. Alternatively, macrophages are very important for clearing dying cells from the body. And a macrophage that's eaten a cell that's undergone apoptosis will polarize in a different direction, so-called M2 macrophage, which is really involved in tissue repair, but these macrophages also suppress immune responses. And in the tumor microenvironment, the majority of the tumor-associated macrophages have this phenotype and suppress immune responses. So Larissa began to do experiments. She knocked out the PI3 kinase itself in myeloid cells, including macrophages. She knocked out the E1, part of the E3 and the E2 ligases, respectively, on this figure. And in each case, she impacted the number of these little lung modules that would form from the injection of the B16 melanoma. At that point, I said, it's great. You basically have a fantastic observation. It's genetically robust. Autophagy is very important in those in that tumor microenvironment, such that when you disrupt it, you improve the outcome of a cancer. And then she did what really good postdocs do, but sometimes PIs don't like. She did another experiment. And in this experiment, she knocked out a component of the serine kinase complex that is absolutely required for canonical autophagy and could see no effect. I'll show you more examples of this in a bit, but no matter how many times she did this experiment, she could not see it work. And so she had to ask, when is autophagy not autophagy? Now, she approached the problem, we brainstormed it, we thought of certain things, we tested null hypotheses, and it didn't work out. Indeed, this component was gone from the macrophages. There was no autophagy in the macrophages, and yet she could not see the anti-tumor effect. And of course, I just showed you that movie because it makes me chuckle. I hope somebody out there is chuckling as well. And then we thought about some work that had been done about 10 years earlier in the lab. Miguel San Juan is now a project leader at Bristol Myers Squibb and very successful. Back in 2007, he had made a really interesting observation. Miguel took macrophages that were expressing a fluorescent LC3 as a fusion protein, and he fed them yeast particles, which here are in red. And you can see as these come into the macrophage, as they're phagocytosed by the macrophage, the LC3 gloms around that phagosome. It's very rapid. This was happening much more rapidly than we could observe autophagy in any sort of settings. We subsequently found that the LC3 was actually directly lipidated on that phagosome membrane. And as a consequence of that, Miguel had found, in this case, the yeast particles are blue, the LC3 is still green, and the red are lysosomes. And as you can see, as it comes around again, as the macrophage eats a particle, there's the LC3 ring, and those lysosomes stream to the phagosome, fuse with it, and acidify and digest the contents. That's phagosome maturation, which is being facilitated by this LC3 lipidation. At the time, Miguel went to publish that paper in the 2000s, in late 2000s. All the reviewers said, we're missing something. Autophagy is, at that point, was defined by these double membrane autophagosomes that could be seen in electron microscopy. We must be missing them. Something's wrong here. But no matter how hard he looked, he could find no evidence of canonical autophagy, but LC3 was being lipidated on that phagosome, single phagosome membrane. He called it LC3-associated phagocytosis, or LAP, and subsequently found it's not the same as autophagy. Over the ensuing years, we explored that at a genetic and biochemical level, and we still are. But what we found, as did others, I especially want to give a shout out to Oliver Flory and Mike Overholzer, who found that the serine kinase complex was not necessary, which is absolutely required for autophagy. None of the components in that complex are required for LC3-associated phagocytosis. Subsequently, we found that while the lipid kinase itself is the same in both, the cofactors are different. The complex is distinct. And so a protein called AGG14 is absolutely required for the lipid kinase activity in canonical autophagy. It's completely dispensable for the lipid kinase activity in LAP. And in contrast, another protein, Rubicon, which we found by a proteomic approach, is completely dispensable for autophagy, but is absolutely required for the lipid kinase activity in LC3-associated phagocytosis. And while we found that the components of the ligase pathway are the same, again, it was Oliver Fleury who discovered that a mutation in one of those components completely disrupted LC3-associated phagocytosis while having no effect on canonical autophagy. We'll exploit that observation towards the very end of this presentation. But what this did is it gave us a toolbox. We have proteins that are required for both canonical autophagy and LC3-associated phagocytosis. We have proteins that are required for autophagy but are dispensable for LAP. And we found some proteins that are required for LAP that are dispensable for autophagy. We don't have as many of those, partly because we really haven't had screens up until very recently, and I'll mention that as we move along. Now, a few other factoids about LAP. LAP's engaged by phagosome cargo that engage surface toll-like receptors, such as TLR1-2, TLR2-6, and TLR4. For aficionados, this is independent of the usual signaling that comes from these receptors. We now think we know what the signal is, but I'll talk about that next time. LAP is also engaged by other stimuli. FC receptor engagement can do it, and as I'll show you in a moment, the engulfment of dying cells, which will be important in this context. LAP does not require the pre-initiation serine kinase complex and uses a different PI3 kinase complex requiring this protein Rubicon. It's not autophagy. And LAP promotes phagosome maturation, but as you'll see, it does other things as well. Miguel left the lab. Jennifer Martinez joined the lab. She's now a group leader at the National Institutes of Environmental Health Sciences. And Jennifer was interested in the role of LAP in efferocytosis, the engulfment of dying cells. One of the things we do in the lab is kill cells in various ways. It turns out it's not particularly important how a cell dies. It'll be eaten by a macrophage. And in each case, you can see that fluorescent LC3 glomming onto that phagosome, conjugating to the phagosome that contains that dead cell corpse. And that has consequences, much as Miguel had seen with the engulfment of yeast and other things. In the top row, there's a dead cell that's being engulfed by a macrophage. There's that ring of LC3. Within an hour or so, that cell is being digested. We can see acidification as that phagosome fuses with lysosomes, and ultimately, by 90 minutes or 2 hours, all trace of that dead cell is essentially gone. When we disrupt LAP, in this case by removing the E1 ligase, the cells are engulfed, the dead cells are engulfed, but they persist and they persist. And in this little sort of toy experiment, a culture of macrophages was fed labeled dead cells. We looked to see how long it would take for them to be digested. Within about 4 hours, all trace of those dead cells were gone. But in the absence of LAP, they persisted throughout this time. And that changes the biology of the macrophage. So macrophages that eat dying cells don't engage an inflammatory pathway. They don't make inflammatory cytokines. These are a variety of knockouts, bone marrow-derived macrophages engulfing dead cells from a variety of mice. I won't go into the details. In the box are all the genotypes that disrupt LAP. Some of the others disrupt canonical autophagy. In the wild type or the cells without canonical autophagy, there's no inflammatory response, but in the absence of LAP, the cells make inflammatory cytokines. This is showing IL-1 beta. We have the same data for interleukin-6. In contrast, though, when a macrophage from a wild-type animal or from us eats a dying cell, an apoptotic cell, it produces the anti-inflammatory cytokine, interleukin-10. And that production of interleukin-10 is dependent on LC3-associated phagocytosis. We can interrogate that more deeply. This is just a typical RNA-seq. We're now doing this much more extensively using other approaches. But right away, we could notice that there are distinct differences when, in this case, cells with or without the protein Rubicon required for LAP but not required for autophagy, when those cells eat a dying cell, they show a different gene expression pattern. And when we interrogate that, we see enrichment for various inflammatory responses, TNF signaling, interferon responses, but also genes involved in hypoxia and glycolysis. A few years ago, Ed Pierce in Freiburg had shown that macrophages that are induced to have an inflammatory phenotype engage this gene signature of hypoxia and glycolysis. A few more things we learned about LAP along the way. It's involved in the vision cycle. I'll show you that in a moment. It's involved in innate host defense, and we showed that initially, but now many other labs are following up on that. And it suppresses inflammation in a lupus erythematosus-like disease. Just very briefly, in collaboration with Tom Ferguson, we deleted genes required for LAP in the retinal pigment epithelium, the layer of cells that overlay the retina. Every morning when we wake up, we shed the outer segments of our photoreceptors from the day before. These are engulfed, and as that happens in the retinal pigment epithelium, LC3 is conjugated to the phagosomes of those engulfed photoreceptor outer segments, and is required to strip away the spent vitamin A so that it can be reprocessed, made into its active form, which is then redelivered to the retina. That is the vision cycle, and does not occur in the absence of LAP in the retinal pigment epithelium. Our LAP deficient animals lose their vision. As the animals age, when they get to be a year or older, they start to show a mild lupus phenotype with autoantibodies resembling those of lupus patients. This is deposition of immune complexes in the glomeruli of the kidney. And in green or blue, I guess you can see the pointer, in the absence of LAP, we're seeing immunoglobulin deposition in those LAP deficient, but not in autophagy deficient settings. And similarly, deposition of complement component C1q, which is engaged by immune complexes. It's involved in innate defense. Animals lacking LAP, when given aspergillus, in the lower part, you can especially see that the aspergillus is more fulminant at early time points and a little bit later time points in the LAP deficient animals, which don't have LAP within the myeloid lineage. Also, interestingly, they're forming these structures, which are granulomas. And curious to us, was something we haven't followed up on, but perhaps that's another hint as to functions of LAP in suppressing granuloma formation. So back to Larissa. She found that components required for autophagy and LAP produced her anti-cancer effect in her tumor model, whereas a component required for autophagy but not required for LAP had no effect. So she looked in the tumors. In this case, she used tumors that express a membrane-bound fluorescent M-cherry, so they're red. The LC3 is transgenic in these animals, so is green. And the macrophages were identified by their expression of CD11b. You can easily see macrophages that are engulfing dead tumor material, and within those macrophages is nice evidence of these LC3 rings. LAP is occurring spontaneously in tumors. So she knocked out various components within the myeloid lineage, knocking out components of the ligase pathway that are required for both LAP and autophagy. She saw the anti-cancer effect in this model. Knocking out components of the serine kinase not required for LAP, no effect. Knocking out a component of the lipid kinase pathway not required for LAP but required for autophagy, no effect. But knocking out components that are required for LAP but not required for autophagy, she again saw the anti-cancer effect. She saw the same thing in other tumor models. This is just one of them, Lewis lung carcinoma, seen it in a number of other lines, and in genetically modified animals, which form spontaneous tumors. I won't take you through all that data as it's been published. Just another example here. No effect of removing canonical autophagy unless it removes LAP, delay growth of flank tumors when LAP is deficient. So she used single-cell RNA-seq. This was actually the first time we did single-cell RNA-seq of macrophages in the tumor microenvironment. At that time, although we could do the single-cell RNA-seq, the tools to analyze it weren't yet available, and we spent many months working with computational biologists to develop those tools. Now, of course, you can just plug them in online, a lot easier. But the simple message here was that our LAP-deficient macrophages in the tumor microenvironment really had a distinct gene expression signature, which we could see both in monocytes and in different forms of tumor-associated macrophages. And when we interrogated those in the macrophages, one of the features of that different gene expression signature was an interferon signature. So Larissa knocked out type 1 interferon receptor from the animals, which also lacked components required for LAP in the myeloid lineage. Again, we could see an anti-cancer effect in the LAP-deficient animals, but removal of the type 1 interferon receptor completely obliterated that effect. And in addition, when she interrogated that tumor microenvironment, looking at T lymphocytes, she could see while the numbers of T lymphocytes were not different, they were more activated, expressing, for example, gamma interferon and granzyme B, the effector molecule of cytotoxic T lymphocytes. These cells were not deleted for the gene of interest. It was an effect on the macrophages in the myeloid cells. And that increase in activated T cells was dependent on the type 1 interferon receptor. But perhaps most importantly, it was the T cells that were mediating the effect, using her different knockout models, again seeing an anti-tumor effect, a delay in tumor growth in the LAP-deficient animals when she depleted T cells, either CD4 or CD8 T cells, using ablative antibodies. All of those benefits disappeared. And that allowed a few conclusions. A variety of cancers grow more slowly in animals in which myeloid cells lack LAP. Tumor-associated macrophages in LAP-deficient animals may be skewed to a M1-like phenotype with an interferon signature that's likely to be required for this effect. Dying cells also likely to contribute to this effect. I didn't show you all of that evidence. And tumors in LAP-deficient animals show increased T cell activity, without which T cells grow as in LAP-sufficient animals. I'm going to pause there and see if anybody has any questions just for a minute or two.
Doug, I think if you want, we can just continue to the end and then have a question and answer session at the end that'll be like based on how much time we have left.
Okay, yeah, we did lose some time, so I will hurry. Now I'm going to change gears because around this time, Brad Heckman joined the lab. He just left the lab 2 weeks ago to set up his own lab at University of South Florida in Tampa. Brad wanted to study the role of LAP in a different inflammatory disease, which he chose as Alzheimer's disease. And we'll get into why that might have been. It had been shown for several years that the expression of autophagy genes decline with age, and that this decline is more pronounced in the brains of Alzheimer's patients. But Brad asked, could it be LAP instead of autophagy per se? Here's a hint. It wasn't. It was something else, another non-canonical function of the autophagy proteins. The model he used was called the 5X FAD model. These animals carry two transgenes. One of them is for the precursor of the A beta peptide that forms the plaques in Alzheimer's disease. This APP, this Alzheimer's, this A beta peptide precursor was carrying two mutations found in familial Alzheimer's disease, as well as presenilin-1 transgene. This processes the APP into A beta. And again, this was carrying two mutations seen in familial Alzheimer's disease. Well, this is one of the better murine models of Alzheimer's disease that has some problems. First of all, plaques begin to appear after about 6 or 8 months, or become prevalent around that time, at which point you can start to see activation of microglia, which is a feature of Alzheimer's disease. Microglia are the only immune cell resident in the brain. They're a macrophage-like cell, but they are self-renewing, unlike our peripheral macrophages. They don't show any cognitive impairment until they get to be about a year old, and even then it's pretty mild. There really isn't much neuronal loss, as is seen in Alzheimer's patients, although some has been described in this model when animals get to be a year and a half old or so. But one feature that's absent in this model, the phosphorylation of a protein called tau, which is often used as the gold standard of human Alzheimer's disease. This phosphorylation of tau is not really seen in this model. And we'll come back to that. Brad crossed them with different strains. He crossed them with a strain in which both LAP and autophagy are deficient in the myeloid lineage, including microglial cells. Autophagy was deficient, but not LAP, or LAP was deficient in the whole animal, but he also, as I'll show you, did this specifically within microglia, but these animals have intact autophagy. When the animals got to be a few months old, he just couldn't wait. You know, he joined the lab, he did the crosses, he was anxious to do something. He asked if he could kill a few of the animals and take a look in the brains. This is looking in the hippocampus, that's the dentate gyrus. The wild type animals carrying the transgene really didn't show any A beta deposition, a little bit perhaps. The autophagy deficient animals that have LAP didn't show it either. But the LAP deficient animals at this very early time point of just 4 months had really striking increases in A beta deposition in these in the brains, not only in the hippocampus but also in the cortex. That's some quantification. Also showing to the right, these are animals where the knockouts were specifically as close as we can get to specifically in microglia. This uses a inducible Cre, tamoxifen inducible Cre, knocking it out in macrophages and microglial cells, but then waiting 6 weeks, the macrophages completely reconstitute with the gene expression, while the microglia, being self-renewing, do not reconstitute. So these are basically microglia specific. And again, only the microglia specific LAP deficient animals showed this phenotype in that 5X FAD background. So we started to look at microglia and whether they do LAP. He took a microglial cell line, BV2, fed them with A beta, oligomerized A beta, which is in red. You can see that as it's taken up, there are these nice LC3 rings that form around it. It looks like LAP. That deleting canonical autophagy has no effect on that, but deleting LAP completely eliminates that LC3 lipidation, around those vesicles containing the oligomerized A beta. And that's quantified here. But then he ran into a problem. Phagocytosis is defined as a actin-dependent process. There's a drug, latrunculin, that prevents phagocytosis. Indeed, when you take these BV2 cells and feed them with killed yeast particles, that phagocytosis is blocked with latrunculin as expected. Dextran, which is taken up by endocytosis, is unaffected by this as actin is not involved. But the uptake of A beta similarly showed no effect of latrunculin. It's not phagocytosis, it's endocytosis. A couple of months after Brad published this, another group published the same findings, that A beta is taken up in microglia by endocytosis. Here you can see this in a little more detail. Clathrin association, which is associated with endocytosis, though whether it's clathrin dependent or not is not clear, but the uptake is associated with clathrin. The LC3 is on those endosomes, sorry about that. And also this endosomal marker RAB5. But then he ran into another problem. If you recall, both Miguel and Jennifer had shown that LAP is required for this phagosome maturation. Brad interrogated this using a pH-sensitive dye, pH rhodo, on zymosin. As it's taken up, those phagosomes acidify, and that does not occur in the absence of LAP. While they're still taken up to the same extent, those phagosomes don't acidify in the absence of LAP, as Miguel had shown years previously. However, when beta amyloid was similarly labeled with pH rhodo, it was taken up, but the acidification of those endosomes proceeded irrespective of LAP. This doesn't look like it's LAP. And that's quantified here. Further, the degradation of A beta within the microglia proceeded regardless of the presence or absence of any of these requisite genes. So we had a problem. Again, I could show the movie of falling on our faces in the mud puddle. We didn't know what to do, and I did what I usually do. I gave out and gave talks on it and said, we have no idea what's going on. Now, I thought I was terribly clever coming up with this, but some months later, I found a note in the pocket of one of my sports jackets coming back from one of these meetings where somebody, and I'm sorry, I don't know who, if you're out there, please email me because I'd like to give you credit because you put us onto the right track. They pointed out that we should look at this paper from Tony Wisqueret. Tony Wisqueret's lab had shown that components of the autophagy pathway, which are also required for LAP, LC3-associated phagocytosis, influenced another phenomenon, which is the recycling of receptors, surface receptors that internalize into endosomes and then reappear on the cell surface. We used his assay and confirmed what he had found using receptors for A beta, for the A beta amyloid. The real receptor is probably TREM2, but literature also suggests that CD36, a scavenger receptor, and perhaps TLR4, one of the toll-like receptors, are also involved in the uptake of A beta. In each of these cases, the recycling assay that Tony Wisqueret's lab had developed showed deficiency in the recycling of these receptors when the genes required for our non-canonical functions and LC3-associated phagocytosis were absent. We also found that the LC3 lipidation is also required. We did this in a couple of different ways. HEG4 is a protease that's required for preparing LC3 for its lipidation. A dominant negative form of this blocks that and also blocks receptor recycling. Also, this enzyme RABZ, Craig Roy at Yale had shown that Legionella have this enzyme called RABZ that specifically cleaves lipidated LC3, preventing autophagy. It also prevents this recycling of these receptors. The lipidation of LC3 on that endosome membrane is important for that receptor recycling. So we could no longer call it LC3-associated phagocytosis. It's not phagocytosis. It's not involved in phagosome maturation. In this setting, it's involved in receptor recycling of endosomes. So with much thinking about it, we decided to call it LC3-associated endocytosis, or LANDO, having absolutely nothing to do with people named LANDO. So thinking more about this receptor recycling, Brad did a simple experiment. He had seen that the uptake of A beta is unaffected by ablation of any of the genes required for LANDO. However, he asked, what happens if he gives them a second challenge with A beta that's just labeled with a different fluorophore? And there he could see that secondary uptake of A beta was impacted by the absence of LANDO, perhaps because of this defect in the recycling of the receptors. But as with LAP, there were consequences. Oligomerized A beta is somewhat inflammatory. You can see in the white bars that when it's fed to microglia, in this case a microglial cell line, BV2, there's a little bit of inflammatory cytokine production. And this is greatly exacerbated by ablation of genes required for LANDO, but not for autophagy. When he looks in the brains of his 5X FAD LANDO deficient animals, again, he sees upregulation of these inflammatory cytokine genes, but also the marker IBA1 upregulated and activated microglia. And when he looks in the microglia using that IBA1 marker, those LANDO deficient animals carrying those transgenes showed pronounced microgliosis at this very early time point of 4 months, normally not seen until at least 8 months in these animals, and not to this extent. And that's quantified here. Now, I also mentioned this protein tau, which is hyperphosphorylated in Alzheimer's disease. Tau, in its phosphorylated state, destabilizes microtubules. And of course, microtubules are extremely important for axons. And so it's thought that the hyperphosphorylation of tau by disrupting microtubules really impacts the health and function of neurons. And as I mentioned, it's not seen in the 5X FAD model, even at very late times. But this, again, is looking at 4 months and no evidence of that tau phosphorylation, either in the wild-type animals or the animals deficient only in autophagy, but not LANDO. However, in our 4-month-old LANDO-deficient 5X FAD animals, pronounced tau hyperphosphorylation was evident in those same regions where it's found in Alzheimer's disease. That's quantified here. In addition, we start to lose neurons at 4 months. Just the numbers of neurons in the dentate gyrus are decreased just by counting. And also when we look at them, there's increased apoptosis as detected by cleaved caspase 3 within the neurons of this region. So that should have some behavioral consequences. Now, Alzheimer's patients show a condition called anhedonia, a failure to really perceive pleasure. Favorite foods are no longer particularly of interest. They can taste them, they don't really care. Same for favorite people or favorite activities. How do you test this in a mouse? Well, mice like me love sugar. If you give a mouse a choice between water and sugar water, they'll drink the sugar water 95% of the time. You keep switching the bottles around, they'll find the sugar water. You just quantify how much of the sugar water versus the normal water they drank. Sucrose preference test. Even at 2.5 months of age, the Rubicon deficient 5X FAD animals are showing a loss of this sucrose preference, which is completely gone by 4 months of age, where we can start to see our conditional deletion in the E3 ligase are catching up to these, probably because of the efficiency of the CRE being a slight delay. No effect at all of deleting canonical autophagy from the microglia. Similarly, of course, Alzheimer's patients have trouble with directions. Mice put into a very simple maze, the Y maze, will explore it by alternating their exploration between the arms of the maze. When Brad said he wanted to do a Y maze test because it's really very simple to do, he said, you know, good news, it's really easy to do. Bad news is to buy a Y maze costs about $1,000 from a vendor. And I said, this is complete insanity. You know, this is nuts. We should just make one. We asked our engineers if they could make one. They said yes, but you know, we'll have to pay for it. I asked how much, and they said about $30. So I reckon we could probably start a little business selling Y mazes for, I don't know, half the price of other places and still make a really good profit. Actually, we could sell it for a tenth of the price and make a really good profit. Anyway, the I just have to mention that because it's just so outrageous. That spontaneous alteration exploration is lost in our 4-month-old animals where LANDO is deficient in the microglia. The animals have a tendency to just walk back and forth or re-explore the same arm. Also, mice like me like new things and spend more time with new things than old things. It's a very simple test, the novel object recognition test. You give the mouse a couple of objects and let him get used to them over a day. And then the next day you swap one of those objects for a new object and you monitor how much time do they spend with the old object versus the new object. And they really have a great preference for that new object. But in the absence of LANDO, in the absence of LANDO, they lose all discrimination for the new object. They just don't remember. So A-beta is engulfed by microglia and induces LANDO rather than LAP. LANDO functions in the recycling of A-beta receptors. Microglia lacking LANDO show enhanced inflammatory activation in response to A-beta. In vivo, the deposition of A-beta, expression of tau and phosphotau, and microglial activation are all accelerated by LANDO deficiency in the 5X FAD model. These effects appear to have profound consequences for neuronal survival and cognitive function. Expression of genes in the autophagy pathway decline with age, and this decline is more pronounced in Alzheimer's disease. These genes are also involved in LANDO. Is it possible that just a deficiency in LANDO could drive Alzheimer's disease? Now, we're running out of time because of all the technical problems, so I'll go through this very quickly, but please feel free to ask about it. Indeed, we confirmed using samples we obtained from Alzheimer's patients that genes of interest, Rubicon, and several of the other genes in the LANDO pathway are downregulated in the brains of the Alzheimer's patients. But we focused in on this gene, ATG16. ATG16 is a rather large protein. It has a huge WD domain. But interestingly, in some organisms with perfectly good autophagy, that WD domain is not there. And that led, and sorry, there's a typo here. This is ATG16, not ATG5. Tom Weilman was producing a mutant in ATG16, and almost by accident, one of those CRISPR accidents, he inadvertently put in a stop codon that prevented the expression of the WD region of ATG16, this component of the E3 ligase. Those animals were born fine. They had perfectly good autophagy, but subsequently, Oliver Flory, together with Tom Weilman, showed that this was required for the lipidation of LC3 on single membranes, such as in LAP, and we subsequently showed the same was true for LC3 lipidation in LANDO. Tom Weilman wrote me an email and said, I know you're interested in these animals. Would you be interested in aged animals? He had mice that he had been aging between 1 and 2 years. So he sent those animals to us, which was absolutely fantastic. And Brad began to examine those animals. There is no transgene. It is a single stop codon. They are all litter mate controlled. When Brad began to examine these animals, the wild type litter mates looked perfectly normal. These are 2-year-old mice, but the HEG16 mice lacking the WD region had A beta deposition, interestingly, not in plaques, but instead just in aggregates. And yet, you'll see some of the other effects that manifested. Also, activated microglia. We'll look at that in a little more detail. The microglia have this activated phenotype rounding up, as is seen in microgliosis, whereas the wild-type microglia were perfectly normal. Also, looking within that CA region of the hippocampus, the CA3 region of the hippocampus, a lot of tau phosphorylation spontaneously in the brains of these animals. In addition, inflammatory cytokine expression was elevated in the brains of these mutant animals, and that resulted, corresponded to increased apoptosis within neurons and a loss of long-term potentiation across the hippocampus. Neuronal firing was defective. So Brad examined the behavior, again, in the sucrose preference test. Not only did these mutant animals have no sucrose preference, they actually had a sucrose aversion. This form of anhedonia, or almost an antihedonia, is seen in Alzheimer's patients, quite dramatic in these mutants. Complete loss of spontaneous alteration in the Y-maze test, and a complete loss of novel object recognition. So in this animal, there's amyloid deposition, as is seen in Alzheimer's disease, but not amyloid plaques. And a lot of people are questioning the importance of plaques versus deposition of A beta in Alzheimer's disease, which I think our animals are somewhat speaking to. Reactive microgliosis is seen in Alzheimer's disease. Tau hyperphosphorylation, quite dramatic in these animals throughout the hippocampus and in the cortex. Neuronal cell death, clearly evident, and severe behavioral effects in these mice. We've got a model of Alzheimer's disease by a single mutation in the LANDO pathway. Now, based on a few considerations that were starting to emerge, we wondered whether it was the neuronal death or the microglial inflammation that was important here. There had been indications, a Nature paper using a tau transgenic model of Alzheimer's disease, that inhibition of inflammasomes responsible for the production of interleukin-1 were important in the disease process. Brad treated animals with a inflammasome inhibitor, an NLRP3 inhibitor, just for 8 weeks. These animals were already diseased and defective based on their litter mates, but 8 weeks later, microgliosis was decreased, phospho-tau was effectively gone in the brains of these treated animals, and all of their behavioral effects were improved. Despite the neuronal loss, it looks like it's the inflammation that's predominantly mediating these behavioral abnormalities and these memory defects. So with that, I'm going to stop. We're really out of time. I apologize for all the technical problems. These are the people who did the work. I mentioned them along the way. Quick shout out to Stas Sekarenko, who helps us with the long-term potentiation studies, and I will stop there.
Great, thank you for that, Doug. That was an amazing set of stories that you shared. Thank you for that. I'd like to start off with the questions, if possible, especially going back to what you were describing with the LAP that's associated with macrophages that are engulfing dead cells within tumors. And in particular, I was curious as to whether you've seen evidence of LAP hyperactivation or just enhanced activation within certain cancer types or certain tumors that would then promote a more M2 phenotype production of IL-10 anti-inflammatory signaling in order to then, as a means of helping to ensure that the tumor is able to survive? Is there that kind of adaptation that you
That's a
can?
great question. I am extremely interested in this. As you know, studies on macrophages of the tumor microenvironment and different tumors is only beginning to emerge as people use single-cell RNA-seq to analyze the tumor microenvironment. We're very interested in looking at those data as they emerge to see if there's potentially some effect of LAP in those signatures. In addition, we've actually I've reached out to several people, but without much interest. I would certainly be interested to just quantify LAP in tissue sections and see if we could develop the right tools to do that. Since we know, at least in our mouse model, that it's occurring all the time, I suspect that we should be able to use antibodies to detect LC3 lipidation of phagosomes that within macrophages that might have taken up some dead tumor material. Shouldn't be that hard to do. And of course, we're particularly interested to see how this correlates with outcome. So far, I don't have any takers. Maybe somebody listening might be interested to embark on such an enterprise.
In addition, you mentioned that the tumor suppressive effect of LAP dysfunction requires T cells. Have you been able to delve further into exactly how they're involved? Is it via some canonical pathways, or again, something non-canonical?
Yeah, we've spent a lot of time to see if LAP somehow constrains antigenic cross-presentation. This is how peptides from, for example, the tumor might come to be expressed on dendritic cells in the context of class 1 MHC to present to CD8 T cells. So far, we really haven't seen any role there that LAP constrains that, although it would make sense to me given its impact on phagosome maturation. That said, what we have found is that the interferon signature that we're seeing in the LAP-deficient macrophages that engulf dying cells, this is completely dependent on STING. And so now we're particularly interested in why LAP constrains STING per se, and how that might work.
That's an interesting observation there. We have time for one more question. So somebody wants to know, considering that there are a myriad of different models for Alzheimer's disease, have you been able to look at the effects of LANDO dysfunction in the acceleration of AD-associated phenotypes in other models? Like you showed at the very end that basically knocking out or causing LANDO dysfunction on its own is capable of driving this process. But in models where you do see more neuronal cell death throughout the development of the symptoms, will the loss of LANDO accelerate that process?
Yeah, certainly we have interest in looking at some of these. One is the Indiana mutation, and another is the so-called 3TG model of Alzheimer's disease that's a little more robust perhaps than the 5xFAD model. Brad has setting up his own lab, and that is definitely in line with some of his interests. We're going to step back a bit and really try to interrogate, using our two models, a little more of the mechanism and whether, for example, there are roles for interleukin-1 specifically in some of these disease processes, and where those interleukin-1 receptors might be important.
Very good. Unfortunately, we're out of time, Doug, but we really do appreciate you coming and speaking today. It's a great pleasure, and of course, always a treat to hear about your latest research. So thank you again, and we look forward to seeing you next time out at one of the many that you attend.
Absolutely, Chris, and thanks for having me. I will ask people, because of the technical problems and we couldn't get into questions, if you have questions, please email them. You can either email them to me directly, or you can email them to the Dana Farber site. where they'll forward them to me. Either way, I welcome your questions and look forward to some discussions of this.
Fantastic. Thanks again, Doug.
You bet, Chris. It was a pleasure.